The overall goal of the following experiment is to measure rates of DNA repair in cell nuclear extracts in real time using molecular beacons that fluoresce once they are repaired. Base excision repair molecular beacons are comprised of deoxyoligonucleotides in a stem loop structure containing a single base lesion with 6 carboxyfluorescein moieties conjugated to the 5' ends and dabsil moieties conjugated to the 3' ends. When folded, the 6-fam moiety is quenched by dabsil in a non-fluorescent manner via Forster resonance energy transfer. To assess DNA repair activity, beacons are incubated with cell nuclear extracts, which contain repair enzymes. DNA glycosylases recognize and remove the DNA lesion in the beacon. The resulting A-basic sites are substrates for AP endonuclease 1, or APE1. Upon recognition, APE1 cleaves the DNA backbone at the A-basic site, allowing the DNA containing the 6-fam to freely dissociate from the hairpin. Detachment from the quencher results in an increase of fluorescence that is proportional to the level of DNA repair and can be assessed in real time using a quantitative real-time PCR machine.
The capacity to repair DNA damage is crucial to every cell and every organism. Among the multitude of cellular DNA repair pathways, base excision repair targets damaged bases that may result from oxidative or chemical insults. Elucidating the role of individual cellular elements that might influence base excision repair is one of the main objectives in our laboratory studies. As such, measuring DNA repair activity accurately and sensitively has become an interest in our lab. This assay can provide